So good afternoon, everybody. I'm Dr. Lindsey. I'm a senior researcher, part of the TB research group led by Professor Rob Arnouds, located at the Department of Pharmacy at the Radboud UMC. And I want to thank the organizing committee for giving me the opportunity to talk to you today about PKPD investigations and relationships of TB drugs. So in 2019, TB was still the leading cause of death from a single infectious agent worldwide, with an approximately death rate or death toll of 1.4 million, and approximately 200,000 of those were HIV co-infected. In 2020, TB morbidity and mortality will probably be outnumbered, or is outnumbered actually, by the COVID pandemic. But the COVID itself and its measure to contain the pandemic has had devastating impact on TB case detection and reporting in 2020. And it is even estimated that the number of deaths related to TB has increased in 2020, which is showed in the graph recently reported in the WHO report on 2020 TB epidemiology. So you can see here that in TB HIV negative people, there was a rise estimated, and also in TB deaths in HIV positive people. PK or TB drug development and PKPD assessment has been rather slow to develop in TB, in contrast maybe to HIV, as the field that you are more familiar with. Multiple aspects have caused this slow research development and slow drug development in TB. Well, an important cause is a perceived lack of return of investment from pharmaceutical industry that has withheld investment by them in this, it's called a poor man's disease. Then there's TB-specific hurdles that hamper PKPD assessment and drug development. For example, phase 3 trials are really lengthy, with a treatment duration of 6 months and a post-treatment follow-up of 12 months to detect cure-free relapse. Then there is the requirement of TB treatment to be composed of multiple drugs, making it difficult to see the effect and safety of a single drug. Then there is the biphasic treatment of TB with first 2 months of a combination of rifampicin and isoniazid to kill the rapidly dividing bacteria, followed by 4 months of a combination of 4 drugs to kill also the more dormant, more quiescent bacteria. And that makes it difficult to already in early stage predict or see the effect of a new regimen or a new drug. And then there's the TB mouse model, which has been guiding phase 2 and 3 development early, in the recent history, and that has not always proven to be efficient in predicting regimens that would do well in phase 3, and also especially shorter than 6 months regimens. You can also see here that there's been a significant gap in about 30 years in the development again of already existing agents and new agents against TB. So this is something I propose to be the scope of today, and I will explain why our group believes that PKPD relationships and optimal study designs will help elucidate maybe the mechanism of the drug, will help identify optimal exposures to strive for, and thereby help optimal implementation of your drug of interest. So this is all backed up or by this scheme, wherein the drug concentration is the central marker for measurement, and it is the intermediate between the drug that's actually taken and the effect that can be expected. So it can either be positive, so effectiveness, or negative, and that would be toxicity. And the drug concentration as a result of the pharmacokinetics following the ingestion of a certain dose are the intermediate. And this is the area that should be sampled in your study designs. So this is another way of showing the same relationship actually. So here you can see a classic exposure response curve. A certain exposure will result in a certain efficacy, and a higher or overexposure result in a certain adverse effect. And the optimal in between would be the optimal dose for your drug of interest. And clarifying this for your drug of interest, for the drug to be developed, that's key for optimal implementation and making the best benefit, or taking the best benefit from your drug. So in general, pharmacokinetic studies are performed in various phases of clinical development. In the early phases, the objective is often merely to describe pharmacokinetics. So how does the photo picture of your pharmacokinetic curve look like? What various doses will result in what types of PK curves, so what's the dose concentration relationship? And then in more advanced phases, you will look at what are determinants of exposure. And then finally, so the topic of today, what are concentration effect relationships and how does it look like, and what optimal doses should we strive for? So the outline of today, I will first talk to you about the basic concept of what is optimal PKPD index, what are the components of this ratio, and then what should we strive for. And this is more clarified by the examples I will mention in certain clinical models for assess of first-line TB drugs. And then I will move over to high-dose rifampicin, a topic that our group has investigated in depth. And then I will finalize with various outcome markers for pharmacodynamics applied in especially clinical trial setting. So PKPD index. This represents the optimal ratio between exposure on the one hand and bacterial susceptibility as a measure for pharmacodynamic effect on the other hand. And this is often measured by MIC, so the minimal concentration needed to inhibit the bacteria in vitro often. And the PK is represented by a PK parameter in this index, often AUC or Cmax. And a target can be derived from both preclinical work as in clinical studies. And I will give some examples in the next section. And then important to note is that when interpreting such an index is that one, for example, the MIC has been identified in a protein-free medium, that this can be considered as a protein-unbound MIC, and then this should also probably be compared to a protein-unbound AUC in vivo. Okay, and then particularly or specifically in the case of TB drugs, well, available data from literature suggests that for the first-line TB drugs, efficacy or killing as displayed in MIC is exposure dependent. So MIC, AUC of MIC or Cmax of the MIC should be the index to check for. And this is a bit contrary to what is known about antibacterial drugs, So those drugs against cell wall would normally be considered mimicking time above MIC characteristics, and drugs against intracellular targets would be Cmax above MIC, so either time dependent or exposure dependent. But the anti-plosive bacteria is a bit atypical. It's very slow in its duplication. It's also intracellular, so it resides in the macrophage in the lungs, and it can also be in a dormant state, not dying at all. So it's quite atypical bacteria, and current data suggests in general for first-line drugs that its efficacy is exposure dependent. So now I will give you some examples of PKPD relationships in preclinical clinical models. So this is the result from a hollow fiber model. Hollow fiber model is an in vitro model where a set of tubing is able to mimic in vivo PK profiles and which can be related to, in the same system, bacterial killing. So in this paper, it's reported that when mimicking PK profiles that resemble non-adherence, a non-adherence of more than or at least 60% could result in drug-resistant development. And wow, so that means that, okay, maybe non-adherence is not such an important contributor to emergence of resistance, as well, 60% is really quite a lot. And then another set of clinical trial simulations demonstrated that resistance could be developed with low exposures that could resemble pharmacokinetic variability, rendering people exposed to essentially monodrug therapy, for example, with sufficient low exposures. So such a holorizontal model could really learn us a lot on how PK-PD relationships could look like in extreme situations. Then this is another example, the 2D mouse model. And here you can see that isoniazid really reduces long colony-forming units, so bacterial load in lungs, exposure dependent, as AUC over MIC here on the x-axis plotted, and same as for Cmax over MIC, and you see Cmax are often really correlated, so that makes sense. And you see that time above MIC, so a time-dependent measure for exposure, well, does not really relate that well to bacterial killing. So, and this also teaches us about how to dose and interpret isoniazid PK. Then there is this simple analysis performed in humans. So there are a lot of advanced PK/PD analysis on the market nowadays, but this is an example of a simple investigation. So pyrozinamide exposure was divided in low versus normal. And you could see here that low exposures had a larger proportion of patients with poor treatment outcome, as plotted here on the y-axis. And this was the case independent on HIV exposure. So all people with low exposures performed worse. So this is a simple analysis that demonstrates that exposures matter. Then we move to the specific case of rifampicin and high-dose rifampicin, the implementation and development of high-dose rifampicin, which has been investigated by our group for already quite some time. So rifampicin is the cornerstone drug in TB treatment, and its standard dose of 10 milligrams per kilogram has been on the market since the 60s, and the dose selection at that time was based on cost considerations and fear of especially hepatotoxicity. And recently, I mean, like maybe a decade ago or something like that, groups of people, amongst them also the group here at Radboud GNC, were thinking, okay, maybe this dose is too low, maybe we should dose higher, and higher exposures make us go more higher up in that exposure response curve that I showed earlier. So one of the first populations that we piloted with high dose rifampicin were TB meningitis patients in Indonesia, together with our collaborators in Bandung, a group of Nina Ruslani and Ahmad Ghanim. And we saw that indeed higher doses and more specifically in this analysis, higher exposures resulted in an increased proportion of survival, as you can see in this nice simulation, where you can see that exposures that are associated with the dose of 30 milligrams per kilogram really perform much better than exposures associated with the standard dose, resulting in a survival of approximately 50%, which could be increased to 75% with increased doses. So this is a really nice example of when data analysis of various smaller phase 2 studies can show what an exposure response looks like and support dose selection for this devastating TB disease. Well, another analysis of high-dose rifampicin dosing in pulmonary TB showed again that increasing the dose, increasing thereby the exposure of rifampicin, resulted in better clearance of bacteria in the sputum, so weak 8 sputum conversion is plotted here. The magnitude of effect was less staggering here as compared to TB meningitis studies I just showed on the previous slide. But still, you could see there was this improved effect in the culture conversion, and there could not be, a plateau could not be seen. So it seemed like it was still going linearly up. So maybe higher exposures would even do better. So these are the results recently published last year of a dose escalation study performed in South Africa within the Panacea Consortium, showing the early bactericidal activity, so the decline in bacterial load in bacteria in the first days of treatment. So this was 7-day monotherapy with various higher doses of rifampicin, so it's a dose escalation study. And you could see that the sputum clearance was indeed increasing for higher doses. So you can see the various dosages tested, and the clearance was increasing with higher doses. And then we could also see that here were various number of adverse events experienced, which was also increasing with higher exposures. So somewhere between 50, which caused the largest bacterial clearance early on, and also the highest probability of developing adverse events. Somewhere there should be the optimal dose with the most optimal killing, but the least amount of adverse events. And this will be then, those can be proposed on basis of this modeling for phase 3 testing. And then there's the issue around protein binding, which I just shortly already touched upon with interpreting PKPD assessments. So for rifampicin, we earlier already saw in combination again, or in collaboration again with our Indonesian colleagues, that there is a large variation in free fraction between individuals, ranging from 5 to 15%, which made us hesitant. Is total concentrations, is that the most optimal type of exposure to do PKPD assessments with, as there is so much variation between individuals? So in another data set, here collected again also among the Panacea Consortium, we correlated total concentrations with unbound concentrations and total AUCs with unbound AUCs, and we saw, well, good correlation. And when we did a prediction of unbound concentrations from total concentration, we saw this was done with a really acceptable bias and imprecision. The bias was almost 0%, imprecision was around 15%. So that made us conclude, okay, yes, we can use total concentrations for this drug to do PKPD assessments. Then a question we often get when presenting about high-dose rifampicin is, well, we know rifampicin has a high capacity of causing drug-drug interaction, also with antiretrovirals and antidiabetics, often combined with TB drugs. What are the implications of high-dose rifampicin when it comes on the market and it's combined with all these drugs that are known to interact with or are a victim of interactions when combined with rifampicin? So historically, few data have been available around the 10 milligram per kilogram standard dose, showing that, well, it doesn't impact the amount of exposure of the victim drug that much if it's a higher dose. But that was in the lower end of the doses, around 10 milligrams per kilogram benchmark. But recently, as two studies have been published or has been presented at CROI, I think also last year, that doses of 20 and 35 milligrams per kilogram combined with efavirenz or dolutegravir have only showed a modest impact on dolutegravir and efavirenz exposure as compared to the standard dose, and combination appeared to be safe. So those are the first signals that, yes, it can be combined and it will not clinically significantly impact the drugs as compared to the standard dose already. And to investigate this even more detailed, we are currently in Panacea performing this within subject study design, providing patients with a cocktail of drugs that represent our model substrate for a set of probe drugs for a set of CYP enzymes and a PGP transporter. And this cocktail is given after 2 weeks of standard dose, and then again after another 2 weeks of high dose rifampicin, 14 milligrams per kilogram. And this should definitely give an idea on how exposure dependent the drug-drug interaction of rifampicin are. So results are expected for this year as enrollment is already completed. So then I come to the final part of this presentation. I will discuss some pharmacodynamic outcome markers with you and the differences between the various outcome markers. So because of the lengthy time to the ideal clinical outcome marker, which is the relapse-free cure, is time-consuming and lengthy in a phase 3 trial setting. There has been a lot of efforts in finding optimal surrogate marker in short-term studies that really translate well to outcome in phase 3 studies. So I will discuss a few examples with you today. So some of you may already know about a culture conversion or time to culture conversions in 2 months, which is often used as a surrogate for outcomes of how many people have cleared their bacteria in the sputum at this time point. Then there are just general X-ray results. How do the X-rays look like of the lungs? Then there are currently mRNA-based methods. So these are often based on the qPCR technique, magnifying TB RNA, and thereby concluding something about the presence of bacteria load, actually live bacteria, because they will synthesize this RNA. So this is actually a summary already of what I told you today that long-term relapse-free cure is the ideal marker in the phase 3 setting. And then there are various intermediate biomarkers of clinical outcome given, for example, by early bactericidal activity studies. So bactericidal activity is decline in bacterial load in sputum over a certain time interval, so either 2 to 14 days or maybe prolonged over a set of multiple weeks. And then there's sputum conversion after at 2 months or thereafter. And so regarding the EBA is actually often the first study to be done in patients in TB, but it does not always correlate to long-term effects. For example, if you can see here the graph of bedaquiline performance in EBA, you can see for the highest bedaquiline, which is CMC 207, 400 milligrams, the bacterial load only declined later on in the EBA. This probably has to do with the PK characteristics of bedaquiline that first has to accumulate to reach effective concentrations. So essentially, EBA was very poor of this drug. While later on, data was collected. It was rather effective sterilizing drug. So EBA is not always predictive in that way. But it is an efficient trial type. It is short. You can see the effect of multiple combinations, do comparisons. So it's a popular trial type in TB. Then there is the example of the mid-long marker, sputum conversion. You can either have percentage at month 2, for example, or you can include the longitudinal information and assess time to culture conversion. Still, it's a binary interpretation, so it's either converted yes or no. So it doesn't give continuous information that is available from, for example, the culture, how many CFUs were there. So it's a bit you lose information in this sputum conversion endpoint. But it does seem that month 2 culture positivity relates well with the number of patients, increased patients with recurrence when there is increased culture positivity, and indeed when also seeing specificity and sensitivity of predicting clinical outcome, that culture measures perform better, but only, maybe a little bit better than, for example, from baseline covariates alone, which is this light green line, which is most close to the chance line. But still, it's not optimal. So the various markers used in these study types, just so the techniques used to quantify the bacterial load, most often used are the gold standard, which is historically used most often and has the most comparative data and literature. Those are the colony-forming units. They are measured on agar plates, and colonies are counted as a measure of bacterial load. Then there's the time to positivity in liquid culture. So bacteria are cultured in a tube, and when bacteria reach a certain threshold present, then the tube becomes positive. But both measures take time because they are dependent on the duplication of the bacteria, and that's shown to be. So they are continuous measures, but they take time, and both have quite some comparative data and literature now. So that's a pro. And there are the more modern or the newest techniques that should give a result much faster. So these are, for example, the MBLA assay that's also being developed within Panacea that quantifies TB RNA based on qPCR technique, and that should give results rather quickly. Downside of this technique is that the lab should familiarize itself with qPCR, which can be quite laborsome. So the optimal biomarker, so the optimal marker of treatment response should, well, preferably mimic the endpoints in phase 3, so relapse-free cure, most closely. It should have a dynamic range, preferably so continuously, so that it can be continuously, as a continuous variable, be included in PKPD assessment. And preferably this can be measured early on in treatment. So when comparing two of these markers, CFU and TTP, various literature data are there that show that they are indeed correlated with each other. And the TTP, for example, can substitute CFU also for EBA studies, where CFU is still the gold standard for EBA studies. You can see here that there's a clear correlation with TTP increasing a little bit for the same CFU measures later on in treatment. And you will see that while developing new markers, these will always be compared to first CFU and then also TTP to see how they relate to each other, because these are the gold standards at the moment. So take-home message from me to you is that PKPD methods play an important role in TB drug development, as well as improving implementation of available TB drugs, as I showed for the case of rifampicin, and really optimizing or elucidating this exposure response curve for each drug really helps in implementing the optimal dose for a drug. I can show you now an example of a study that we really designed with as primary objective exposure response of a new drug in TB. So in Panacea, we built on the knowledge of the newest TB regimen, MDR-TB regimen accepted by FDA and EMA, containing bedaquiline, patomanet, and linezolid. We came up with a new regimen, primarily focusing on sutazolid as just an effective drug as linezolid, but with presumed less toxicity, and then combined with a backbone of bedaquiline delaminate as another pretuminide, so a nitro-imidazole sister, so to speak. And then we added moxifloxacin to add sterilizing and bactericidal activity. Well, the background drugs, BDM, as we are most interested in sutazolid, we call BDM the background regimen, has a good safety profile based on earlier phase studies. Well, maybe the highest safety signal, or the most important safety signal, is a potential QTC interval, because they all separately can produce QTC interval prolongation, but it shows there is not, well, it seems there is not that much of a potentiating effect when combined. So when checking the murine data of sutazolid, we saw that sutazolid compared to linezolid with similar backbone cleared bacteria faster in the mouse model, with lower CFU counts in the mouse model for sutazolid as compared to linezolid here, and with similar relapse. So this supports the efficacy of sutazolid as a linezolid substitute. And then we designed this study in which we randomized patients to 5 different arms of BDMU, so bedaquiline, delaminate, moxifloxacin, and sutazolid, of which one arm has no sutazolid, and the other arms have increasing doses of sutazolid. Such a design will give, well, maybe not that much information per arm, but it will give a broad range of sutazolid exposures, and that will enable exposure response modeling. So this is an example of a study that has been really designed to perform exposure response modeling and not just look at one particular dose and its efficacy and toxicity. So objectives, yes, exposure response, exposure toxicity modeling, and while recruitment has been almost completed, so we hope first results are there late next year or late 2023. So I would like to thank all these people here. Dr. Elin Spencer, Professor Rob Arnast, part of the TB research group at the Radboud UMC, and then all the other departments at Radboud UMC, with whom we closely collaborate, internal medicine, microbiology, and pulmonary medicine, and our colleagues in Indonesia and our colleagues in the Panacea Consortium, with whom we perform a lot of these studies, and then Uppsala University, with whom we do a lot of the PKPD analysis. So this was it for me for today.